Understanding the SutterPatch data structure will help us understand the next couple of segments that I will talk about. Structured data obviously make data analysis easier, help you finding your data. The definition of an experiment is relatively open. We recommend that you define the recording from one cell or maybe one preparation, such as a tissue slice, a brain slice, or a cell culture dish as one experiment. Within that experiment, you will record what you may know as a protocol or a pulse generator sequence. Typically a sequence of command steps that you use, for example, to create, as shown here, a sodium IV curve, a current voltage relationship, one or multiple signals, usually with multiple sweeps. And some software leaves you with this as the whole data file, and then it's up to you to determine how this relates to the experiment and also to other routines or the equivalents of routines that you may execute here. We don't like that. That's why we created a container for the routine data for the series, which is called a paradigm data. And that's automatically started when you start a routine. We start a paradigm, and that paradigm contains information that can be automatically determined, such as what amplifier model was it? What was the serial number? What computer was used? What software version was used? Who was logged into the computer? Obvious things like what was the date and timestamp, everything like that. But it also lets you add user-defined information, such as what animal species you recorded from. Was it mouse today? Who prepared the cells, at what time, in what solution, things like that. If it's important for you, you can record what puller you pulled your pipettes on, what pull program you used. But also, and maybe even more importantly, the paradigm keeps track if you execute the same routine again or a different routine, what is the temporal context between the two? And it also lets you set a tag if you want to switch your solution, your bath solution, to something that contains an ion channel modulator, even in between routine acquisition, where you don't even record data point by point. It also keeps track of that if you set a tag or control your solution switcher through SutterPatch software. So all that information is contained in the paradigm, with the goal that eventually you will end up executing what we call planned paradigms, where you have a paradigm here that could be your sodium IV. Then you can have flow control. You can have if, then, else, or loops, for example. In our sodium example, we could determine the half-an activation potential in the second routine that we execute, and then even chain to a second paradigm that executes the data that you can use to create a concentration response curve. So understanding this data structure, the tiers of the experiment, the paradigm data, and the routine data, that's important to understand what we will do next, which is creating a routine like in the previous slide, where we create the data for a sodium IV curve or something like it. Now that we understand the structure of the data that we're going to record, let's switch back to our membrane test. We have our capacitance compensation all on and get a pretty decent signal. We can look at the noise, make sure it has little enough noise. We can directly measure the RMS noise here, 4 picoamps in a 10 kilohertz band. That's actually pretty good considering that we have the model cell without any shielding sitting in a perfectly normal room here. Let's close that and open the routine editor. The routine editor shows you a routine pool on the left, which is a collection of, in this case, sample routines, different waveforms that you can put out and base your actual routines on. The idea of a routine pool is that you have your standard 3 or 5 routines that you use in your standard experiments in a routine pool that might be named something like my standard routine pool something along those lines. The sample routine pool has many more than you would normally have. And on the right of each routine, you see a description of what the routine does. And what we will do now is we will create an IV routine. I call that my IV. And we will create that from scratch. I can't use spaces or special characters in routine names because the routine names get inherited to the data, and the data names can eventually be used in mathematical equations. They will be tripped up by spaces or special characters, hence only the underscore in the name. And let's take a look at the acquisition and routine parameters. What I'll do here is I'll change the number of sweeps to 15. And once I enable that, I can set the sweep to sweep duration to 1 second. And that's all I want to change at this point here. For my input channels, we only have one head stage connected to the DPatch here. We can look at the current. We'll also look at the voltage in this case. And for the output channels, that's where it becomes interesting. The stim out 1, that's the channel that is hardwired to the first head stage. We're going to edit the waveform. We have a baseline of 10 milliseconds in duration. We'll enable the second segment here. And that is where we increment our potential. We start at something physiological, and that is minus 80 M for millivolts. All units are in SI units, in volts. So had I entered minus 80, it wouldn't have accepted that because that would be interpreted as minus 80 volts. The increment value shall be 10 millivolts here. The duration, let's make that a little longer. Let's make that 100 milliseconds. And if I decided, okay, I really don't want to depolarize as far as plus 60 millivolts, I can also change the number of sweeps again here. And you see how the real-time preview down here updates the moment I get out of edit mode. Once I apply that, I have my waveform here. And I enable our tail current segment. We'll make that a little longer, 50 milliseconds, because we may have longer tail currents depending on the cell type. And we can either set that to a fixed value, or over here on the amplifier control panel, if we assume that we have a physiological holding potential of minus 60 millivolts, we can also set this to hold, and then the segment amplitude here is set to the holding potential, and we do the same for segment 1. Now we got a viable waveform already, and we could run this routine. But what we want to do is, in real time, we want to create a current voltage relationship. So we'll move on here to the real-time measurements and graphs. We have up to 16 measurements here. And what we want to measure, since we only have a model cell connected here, we want to measure capacitive transients. We will not have elicited sodium currents, which would be inward humps, but we will have sharp capacitive peaks, most of them outward. So that's what we're going to measure here. And I will call this outward peak. Here, this is only a label, so this can have a space in it. And what I'll measure is the max value. And I want to measure that from current 1 and relative to my segment time. And the segment that I'm interested in is segment 2. This is segment 1, 2, and 3. I'm interested in segment 2. That's my segment with the steps. And I want to start a little bit before the beginning of the segment. So I start at minus 0.022% of the segment duration. You see how the preview updates the measurement region already. And my capacitive transients are relatively short, so I only measure the first 10% of the segment duration here. Then I need my V measurement. That would be my voltage step. And I can either measure that as the mean, again from segment 2. I can measure either the whole segment or something in the back. Or what I can do is, rather than measuring the mean, I can directly measure the segment amplitude because we are putting that out. So we know what that is from, the stimulus out channel 1, the amplitude of segment 2. Actually, let me rename the measurement, and let me call that command step. So now we got our measurements, the I and the V. Let's make sure we create a graph. Let me enable the first graph here, double-click into the label field and call that IV. On the y-axis, I want to plot the outward peak. Labels and units get inherited automatically. Remember, we're working in SI units. That's why this is amps rather than picoamps or nanoamps. We don't have scaling factors. We directly control the amplifier, and we know what units come back, so we don't need to worry about, do we have picoamps or nanoamps today? And on the x-axis, we don't want the time, but we want the command step. And what I'm going to do here is I'll rather put that in as an equation. And the equation in this case is one of the measurements. That is M2. And if for whatever reason that makes sense, I could take that times 15 or minus 5 or whatever. So I can apply any equation, do the math for the creation of the display here. But in our example, M2 should do the job. And if I selected it from the drop-down here, it would automatically inherit the labels. Here, what I'm going to do is I'm going to manually edit the labels. And of course, the unit would also have been inherited. I'm going to manually set that to volts here. And that now should give us a graph that represents the IV curve. Let's do that real quick. Before I hit execute, I hit save pool. With the pool saved, after we made changes, we can execute or activate the routine. The difference is activate sends the definition of the routine, all the settings to the scope window, and execute hits the start button for you at the same time. That's the only difference between the two. And here we see our steps coming in and our current in response. And we're measuring the capacitive transients, the capacitive peaks here, the outward peaks. And the IV curve is created as the data are recorded. Let's do that again. And also watch the VU meters here. This shows you the size, the amplitude of your signal, in this case the capacitive transients, relative to the digitizer range. If it's green, it means, yep, it's good. Your signal is big enough, so you don't see digitizer noise, but it's far enough from saturation levels.
Follow along the next episode in the series, or jump to the playlist for other informative videos. Subscribe now to keep up with the latest installment.